Everybody,
Oh.
thank you for coming. I'm going to stand so it's a little easier to have some energy. I'm going to talk about zooplankton and Waquoit Bay, but more generally about zooplankton. And I was asked, asked to give a little bit of my origin story and how I started working on zooplankton, and it is a little bit of a twisty path. So I grew up in the Chicago suburbs where there are not a lot of zooplankton. It is famously pretty much landlocked. But as I was growing up, I was very interested in the water. My favorite thing was to go to the pool. So I became a competitive swimmer. My highest aspiration as a small child was to be a lifeguard, which I managed to do. So that was great. And then once I was able to go to college, I did my undergraduate work at the University of Miami. I got to do some exchange year in Australia, and then I did my graduate work in Hawaii. So as I was doing that, I did start to learn about marine environments. I became a really avid scuba diver. I focused on coral reef research. I was really interested in how the corals grew and adapted to their environment and the complexity of them. And my graduate work focused on what's called endocrine disruption, the idea that chemicals in the environment that mimic hormones or maybe our natural hormones released in sewage could be disrupting the reproduction of other animals. And so we were looking at estrogens affecting corals. It turns out that even though corals have very different reproductive systems than we do, if they're exposed to estrogens through sewage, it can disrupt their reproduction. And so that's not zooplankton, but it is maybe a little bit of how I got interested in coastal environments and coastal impacts. And then I came to Hui back in 2002. And at Hui, I was able to move much more into lab-based research that was really taking advantage of all the advances in molecular biology and sequencing technology. We were starting to get genomes from these organisms and really understand sort of how their biology ticks. And so, I thought it was more fun to work with animals that are local, that you could kind of mess around with in your backyard. And so I got interested in working with a sea anemone called Nematostella. And so the sea anemones are in the same big group, the same phylum as the corals. So they share some of the same biological features, but they're just interesting because they're local. They live in salt marshes in the tidal pools. So this is Sipawisset Marsh, which is right in Falmouth, and we can dig these things up. They're weird little sea anemones. You might think of, if you watch like Finding Nemo or something, you think about the sea anemones on the reef. These are quite different. They're burrow in the mud. And one of the things that's really interesting about them is they're very tolerant to a wide range of environmental conditions. And so if you think about where they live, these animals are exposed to like a big range in temperature. We know that. We've experienced some big ranges in temperature locally. So over the course of the year, but even over the course of a day in the shallow tide pools, there can be big temperature ranges, big salinity ranges, day-night oxygen changes, and then also all the chemicals that are in the water and the sediments these animals are exposed to. And so we've studied a whole bunch of different aspects of their biology over the years. And I'm just going to give you a little flavor of some of the things my lab has done. So some of the things we've learned about Nematostella, the sea anemone, they have circadian rhythms. So just like we do, you know how you get jet lag and you're tired at night, you're awake in the day. Sea anemones have circadian rhythms too, in their behavior, in their metabolism, in their genes turning on and off. And it seems to be controlled by some of the same genes that control our circadian rhythms. So my lab has studied that from an evolutionary perspective. I told you that those animals are really tolerant of a lot of things. One of the things that's interesting is they're very adaptable. And so we found that, for example, if you take mother sea anemones and you put them in some hotter water, their babies will be more tolerant of the hotter water. And it's a really neat sort of short-term adaptation. And then another example is they don't like ultraviolet light, as far as we can tell. So if we put them in ultraviolet light, like in the sunlight, that really creates a stress response in them. And it also makes them more sensitive to other chemicals. And you might think, well, they usually live in the sediments. So why do they even care? They could just hide. But we really think it is a relevant stress to them because when we look at their gene expression, when we look at what they're doing with their biology, in the morning, they're cranking up genes that help them make natural sunscreens and help them repair DNA damage. So that's sort of the kinds of things my lab was cranking along on for a bunch of years. And I'm really interested in sea anemones, really interested in corals. But one day I went to my department seminar and I saw a seminar by a scientist named Mark Baumgartner, who studies right whales. And he was tagging these right whales and looking at where they're swimming and what they're eating. And what he found is that the right whales, they'd be up at the surface, and certain times of the year, they'd swim down into deep water, and they'd stay at deep water for their whole dive, 20, 30 minutes, and then they'd come back up. And when he was looking at what's in the water, it turned out it was a very special group of zooplankton that were there. And so what we know is that baleen whales, like right whales, they feed on zooplankton. And because the whales are so big, they need a lot of zooplankton. So they need to know where these zooplankton are aggregated. And so what it turned out they were eating is a type of zooplankton, a copepod called calanus, that's really full of energy. It's you can see in this little animal. I think you can see if I move my cursor. Yeah. Okay, let's see if there's like a little coordination test. This little cylinder here. This is where they store fat. So these animals, the copepods, they're eating algae, they're storing it all as fat, and so they're really full of energy. And there's in some places there's a lot of these animals, and in fact, there's so many sometimes that you can see them from outer space. The reason you can see them from outer space is if you look at this little animal, see how it has these little orange parts? It's catching that orange pigment from the algae that it eats. It's accumulating it in its body, and that's what allows them to, under some circumstances, even see these animals with satellites because there's so many of them. So that's cool. So these copepods are really important to the ecosystem. But the part that got me interested, remember how I said they were like in deep water? Okay, so they're in deep water for a couple of reasons. Normally, zooplankton during the day and night, they might move up and down in the water. So often they're up at the surface at night to feed. And then in the day, they don't want other predators to see them. And so they move into deeper water to kind of hide. And so we call that diel vertical migration. So zooplankton do that in the open ocean. They're moving up and down. But these copepods do something extra special. So they also have a seasonal migration. And so what these guys do is certain times of year. So in this plot, the y-axis is depth and the x-axis, is showing you sort of the season. So in certain times of year in the spring, the animals are up at the surface, growing, breeding, eating, doing their diel vertical migration. And then as you move toward the fall, they start running out of food. There's a lot of predators. They kind of say, this isn't such a great place to be. And so basically the teenage copepods, they've been eating and fattening up, and they move into this deep water and they go dormant. And they can stay dormant for up to 9-10 months, depending on where you are in the world. And the part that I think is crazy is they're doing this in the middle of the ocean. They're just floating in this deep water and hanging out until the conditions get better. The other thing that's interesting about that is whales caught onto it. And so the whales are able to go down and basically vacuum them up. And so this dormancy is just a really important feature of the ocean that lets these copepods survive a seasonally variable environment. But it's also really important to where food is in the water column. So that is how I got interested in zooplankton. This is an example of what happens in the open ocean. This isn't happening, in Waquoit Bay, they're not doing this seasonal dormancy. It's too shallow. We don't have this species. But that's how I got interested in zooplankton. And so enough about me. I was asked now to give a little bit of just a introduction to some of the key terms and things we think about when we're studying zooplankton. And so some of you might know this very well, but I think it's fun for all of us. And I was kind of excited that as I was getting ready to start this talk, some of you enjoyed guessing what some of the zooplankton are. So we do have a little bit of that we can play. So zooplankton in general are marine or aquatic animals. So you think about zooplankton, and they're drifting in the water column and they're not able to swim against the ocean currents. So they're either just very small or they're not very good swimmers. They can usually, swim to some degree, but not able to like swim against a current. And as you can see, there are all different kinds of zooplankton. And one of the things that we love to do as biologists is to sort things and classify things. And so I'm going to show you just two of the ways that we classify zooplankton. Does anyone want to guess how we classify them? Well, one way is by size, right? So we have different categories. So one of them, we talk about the microplankton or the microzooplankton, and those are 0.02 to 0.2 millimeters. That's pretty small. So a human hair is about 0.5 millimeter. So this is thick. So this is smaller than the thickness of a human hair. And so some of the things that are that small are some copepod nauplii. So those are like baby copepods can be that small, depending on the species. Baby sea urchins can be that small. And then this isn't an animal. This is so it's not microzooplankton, but it's microplankton. This is a ciliate. So this is a single cell organism with protist. So we have other types of organisms that can be in that size range. Okay, so that's microzooplankton. And then the next size up, which I think is the best size, is the mesozooplankton. So some of the mesozooplankton that we see are pteropods. So these guys are basically oceanic snails. They're really cool. So they have a shell that's made out of calcium carbonate. They have like little, basically wings that they can flap around. And they, depending on the species, they make a net of mucus that they use to trap particles in meat. Does anyone know the common name for these guys? Phil does, but Dan probably does. So these, some people call them sea butterflies. Yeah. And then we've already talked a little bit about copepods. There's all different kinds of copepods, but a lot of them are in this mesozooplankton range. And oftentimes the copepods are the biggest part of the mesozooplankton. So copepods are crustaceans, so they're related to things like lobsters and crabs. They're also related to insects. And then cubing nests, the common name for these guys are arrowworms, and they are terrifying, voracious predators. They have these poison jaws that they capture their prey with, but they're very little, so you don't have to be scared of it. They are scary under a microscope, though. Yeah. And then when we get to bigger sizes, we have the macrozooplankton. So these things are a couple centimeters in the megazooplankton. And so again, that could be a bunch of different things, but I tend to think of things like ctenophores, which are cone jellies, or jellyfish, sea jellies. Some polychaetes can be that big, some worms. Okay, so you might be wondering why we classify them by size. And there's a couple reasons we do this. So one is the size tends to correlate with where they are in the food web, and little things usually eat little things, bigger things usually eat bigger things. The baleen whales are a good exception to that, which are very big and eat very little things. But here we show, microzooplankton, which can be, for example, fed upon by the mesozooplankton, which can be fed upon by some of these large carnivorous zooplankton. So it's sometimes a way of parsing out different trophic groups. And then another reason we do this is it's practical. So one of the main ways that we sample and study zooplankton is using nets. And so the nets usually have some sort of mesh size, and the size of the mesh is going to affect what kinds of organisms you catch. And so it's really a practical thing of how we sample the environment. Okay. And then another way we classify zooplankton, I would broadly say, is by their lifestyle or their life history. And so there's really two main categories that these fall into. So the meroplankton are the ones that spend part of their life in the plankton. Usually, sometimes that's the larval stage. And then part of their life, they'll either recruit to the benthos, they'll live on the bottom, or maybe they just get big enough that they can swim against the currents. And so then we call them nepton. So this is the part where I haven't labeled them, so you can kind of guess. Does anyone know what this ferocious looking guy is? It is a crustacean sometimes, yeah. It is a lobster, yeah. And I think Dan knows what this one is probably. No?
Oh, yes.
Yeah. So this is, does anyone else know what this one? Doberman. Yeah, this is an oyster, yeah. And then these are fish larvae. So we have, this is a spiny lobster larva. This is some oyster larvae. And then, yeah, larval fish. So these guys will eventually become benthic. These guys will eventually become, well, to be fair, I don't know what kind of fish these are, but they would probably become nectin. So they'd be swimming against the currents. So that's the meroplankton. The other group is the holoplankton. So they spend, the way to remember, that one's easy to remember, like hollow is like whole. They spend their whole life in the plankton. And so that's some of the organisms that we've already seen would be the pteropods, like this guy, the cone jellies, the keating gnats, and then this is a different copepod. So these guys more or less spend their whole life in the plankton. There are some like cases where it's like a little bit complicated to think about. And here's an example of a type of animal that we do have in the bay. So this is showing some copepods. So this would be, for example, some of the Centropodes copepods, where the adults and the babies, they're up in the water column, but they can produce eggs, where the eggs can go into the sediments and sometimes can stay there for months or years. And so that's a different type of, you know how when I was talking about the oceanic copepods, where they have that dormancy where as teenagers they're hanging out in the middle of water, this is a different kind of dormancy where they can lay eggs that are sort of like little cysts. And so the eggs can be dormant in the sediments until the conditions are right to stir them up and get them to hatch and germinate. And so that's a really common feature in a lot of shallow waters where we have these dormant stages that become usually with the spring where we get some mixing and sort of conditions favorable to their growth, they start to repopulate the water column. So yeah, I'm just going to talk a little bit about the work that we're starting to do here in Waquoit Bay. And it's pretty new, so I'm not going to show data per se, but I'm going to talk about the approaches we're using and the methods that we've been using. So we became interested in Waquoit Bay because it's a research reserve, and there's really robust ongoing monitoring of water quality and some of the environmental conditions like temperature and like chlorophyll and nutrients and stuff like that. We also noticed that there's not much data about zooplankton in the bay. So the last real report, the published report that I could find was from over 20 years ago. So there was a paper by Lawrence et al. In 2004 where they were looking at seasonality and spatial patterns in salinity and nutrients, looking at nutrient input into the bay and how that helped to structure the zooplankton community. But you know, a lot of things change in 20 years. So we really thought it would be good to start building up an understanding of what the bay looks like now. So that's what we've been starting to do. Our real overall goal is to understand how the zooplankton abundance and composition change over space and time. So we started doing some sampling in collaboration with partners from Webinar. And so we we're just lucky that there's already an established set of stations through the Baywatch program and I forgot what SWMQ stands for.
System-wide monitoring program.
System-wide monitoring program.
Cool.
Yeah. It's great. And so we picked a subset of those stations prioritizing a couple where there are instruments in the water so that we can get more high frequency data on temperature and pH and that kind of salinity. And we did monthly sampling last year from March through July. We're getting ready to start some more sampling, I think next week. Phil, who's in the room, has been one of the main people helping to lead that effort. And we're doing that again in collaboration with the webinar staff. We go out on their boats and they help us with all the sampling. So I'll show you some pictures of that. We picked these stations partly because a couple of them aren't instrumented, and also we wanted to be able to compare sort of this central part of the bay, inshore, offshore, and then this more riverine influenced arm of the bay. So this is the Childs River, and again, kind of more inshore, closer to the river, and then toward the mouth of the bay. And this is how we do our sampling. We load up, we go out in the webinar boat this in March. It's pretty cold, so we're all geared up. By July, we were wearing shorts and t-shirts. But yeah, go out in the boat, and then we use just tow a net off the boat, using the motion of the boat to drag the net through. We have what's called a flow meter on the net. What that does is as it turns, it gives you a measurement of how much water is passing through the net. And so from that, when you collect your plankton, you know, basically you can back calculate the density of the plankton in the water. So you know how many copepods there were per cubic meter of water. And there's a lot. So and then, so we towed the nets behind the boat. At each station, we did 2 tows for 5 to 10 minutes each. We split those tows. One we preserved in formalin, so we could look at the taxonomy and the size. We could actually look at the animals. The other half we froze, so we could do some biochemical analysis that Wally in the back room is going to be helping us with. We're going to be measuring the energy content of those samples. So after we get the net in, we run it through a sieve and rinse it off, and then we basically just scrape the material into these jars. This is the formalin sample. So this is what it looks like when you have a bunch of dead plankton preserved at the bottom of the jar. So we did that. We bring it back to the lab, and then we analyze it. So this is the fancy new toy that we got. So this is called a flow cam. Basically, what you would normally do when you have this kind of sample, one thing that we still do is we measure the volume of plankton that we collect. So we have that index. But then you would take the sample and maybe take a subsample, and you would look at it under the microscope, and you would have an expert like Phil look at it very carefully and basically classify all the individual organisms and count them and process it, and that's very laborious. This is a faster way. So this is an instrument where we can put a sample on this stir plate here. I should do it on the pointer so that people online can see. You can put a sample on this stir plate here, and then it gets sucked through this pump into this instrument where the sample passes in front of a camera a camera with a microscope. Well, yeah, a camera that magnifies it. It gets magnified and photographed. And I will show you what it looks like. And then we save all the images and can classify them. So this is a little video clip showing you what it looks like as the sample is passing through the flow cam. So as the sample is passing through, it's identifying interesting little images. So some of those are actually pieces of plankton. Some of them might be bubbles. Some of them might be pieces of debris. Since it's short, I'll just show you another time. Yeah, so you can see it's a bunch of copepods, some other things. It's pretty cool, right? Yeah. And then we basically have to sort out all these images, and some of it we can do automatically. So we can tell the computer, okay, we don't want anything that's perfectly spherical and black because that's a bubble. We don't want anything that's really tiny and fluffy because that is just a piece of junk. We don't want anything that's very stringy because that is a string. It's like a feather. So we can sort those things out pretty easily. What we've done after that is really just looking at it manually and just saying, okay, that's junk, that's junk, that's junk. We can get better at doing this automatically using artificial intelligence and machine learning. And so that's what we need to put some time into training the computer, teaching it, okay, this is a copepod, or this is a Centropodes copepod, or this is a Karshia copepod. So we have to put time into teaching it that, but once we do that, the computer can do more of the classification for us. But this is what some of the copepods look like. So these are some of the smaller copepods, these are some of the larger copepods. And then these transparent bits are molts. So copepods, in order to grow, they have to shed their exoskeleton and basically move on to a new developmental stage. And so we can sometimes find these empty pieces of their exoskeleton. So those are some of the molts. And just some of the general patterns that we've been finding so far is, as we expected, the copepods are by far the most abundant part of this mesozooplankton. So it's more than 90% of the zooplankton that we're finding are copepods. Based on the sampling we have so far, we found the highest copepod, the highest zooplankton abundance in March. And then as the season moved on, the abundance dropped. And we don't know this, but what we think is, well, let me back up. What we have seen is that when we went out later in the summer is that there's a lot of gelatinous zooplankton. So things like those cone jellies. So we think it's likely that because you have a lot of these gelatinous zooplankton, they're eating all the mesozooplankton. And that's known from some other environments. In fact, some of the times when we went out in July to try and sample the zooplankton, there was so much gelatinous stuff that we really couldn't even put our sample through a sieve. It was just kind of full of goo. So that's a really important aspect of the environment for us to study and think about, whether that gelatinous zooplankton part of the community is changing. And I can show you, since we do have some time, I can show you an example of the kind of thing that I'm talking about. So that's what we know so far, and this is what we want to really monitor, starting, again, this year and going forward, and try and understand how this might be changing over time, how it might vary from year to year. And it's really important and helpful that we have other environmental data to go along with it. And so, for example, we were able to look at the temperature data from last year and compare it to the past 10 to 20 years. And it looks like last year was not a crazy unusual year within Waquoit Bay. The general timing and the general temperature in the water was similar to what we've seen. So, for example, we wouldn't expect the zooplankton to be super unusual because of the temperature or something like that. But we'll be able to keep looking at the zooplankton community in context of the environmental conditions. So what we want to see is, again, looking at how the abundance might be changing, but also the size distribution, the energy content, the timing of zooplankton abundance, how it's changing from year to year, and how it relates to the water quality. I'll make a point that the size distribution would be something that would be hard to keep track of if you were doing this manually under a microscope, because you'd have to measure everything. But with that FlowCam instrument, it measures the size of every particle. So it'll give us the length and width and everything. So we can very easily track the overall distribution of the size of all these organisms. The reason that's important is the size of the plankton really dictates like how much energy it has. The bigger you are, the more full you are of deliciousness, but also what can eat you, right? So when you're thinking about larval fish, the size of their mouth basically affects what they can eat. So if you get changes in the size of these plankton, it'll affect the food availability to the predators. Yeah, so why does this matter? So, basically zooplankton are really important intermediate links in the food chain. They're food for fish and other predators. More broadly, when we think about oceanic environments, they're important for whales, they're important for all kinds of things, eat zooplankton. And when I say they're an intermediate link on the food chain, they're important food for predators, but they're also important grazers. So they're grazing the algae that are growing in our water. And then what we're really interested in trying to study is the potential for changes in the ecology. And so one of the things, I'll show you a slide in a couple minutes, is if we maybe get changes in the timing or abundance of these jellyfish blooms, how would that maybe affect the abundance of zooplankton, of these mesozooplankton that would be important food for fish? And then again, these environmental changes could also affect the size of the plankton, which again would affect their energy content and availability for predators. I'm just gonna skip this slide and come back to it. I just want to show you one example of a really well-documented change in the zooplankton. I know it's a little complicated, but you guys are a really smart audience, so I could talk you through this. So this is an example of a study that they did in the Chesapeake Bay. And there were two different species of copepods, a blue one and a pink one. These are different species of acarcia. And there was a very well-studied predator. It's a comb jelly, Nemiopsis. And so the solid lines are the historic peak timing of these animals. And so there was this species in blue that would become kind of abundant earlier, more in the spring. And then there's this sister species that would be abundant more in the summer. And then what happened was this jellyfish, as the water started warming, the jellyfish, I'm calling it a jellyfish, it's a comb jelly, the ctenophore, it started becoming abundant earlier in the season. And so, you know, a lot of different things could happen, right? So what could have happened, and what happened here is this blue copepod, maybe it was smarter. No, the way this blue copepod responded to the changing environment is it shifted its timing and it started reproducing earlier, becoming, blooming the population earlier. This one didn't, for whatever reason. And so this one really got eaten up by the jellyfish or by the sea jelly. And so there was basically a big decrease in that population. So this is just an example with three species showing how you can have complicated dynamics. You don't necessarily know how they're going to respond to changes in the climate, changes in the environment. And it's really important to have actual measurements, detailed measurements that we can really look at and try and interpret. So we don't have that data for here yet, but it's really important to have time series so you can understand how things are changing. So yeah, this is maybe a little shorter than I thought it was going to be, but I wanted to just say that a lot of people have helped with this. So some of them are in the room. The really key one who's done a lot of this work is Phil Alatello, but also a lot of other collaborators at WHOI. And then we've worked really closely with our partners at Webner. So Megan Terrell and then Theo Collins, who literally taken us out on the boat, driven us around, and also been just a wealth of information about what's going on in the Bay. They're out here a lot, and so they really know how it's changing. As you know, we're kind of going out once a month, and when we come out, Megan's like, "Oh, you know, did you see these algal mats that are floating up? Did you notice this? Did you notice that?" And so it really helps to have that kind of intuition about the environment, and we appreciate it a lot. And yeah, I just want to thank you guys for coming out tonight and supporting local science. And I'd love to talk more about plankton and try and answer any questions. So, thank you. I will just also say that I think there might be some people in Aquatic Brewery who are online. If anyone's still listening, I have stickers for the people in the audience here, but I also left some of the products. So go up to the desk and see if they have any stickers for you.
We've got some questions from online.
All right.
So we'll ask. Jeff asks, how do you deal with gelatinous zooplankton like the comb jellies? And they are not well characterized by net sampling. They have been booming in recent years.
Yeah, so we haven't been trying to quantify the jellies. I think it's a really important thing to do. And so I've really been thinking that we should find a collaborator to work with on that. You're absolutely right that they are not well sampled by nets. They get turned into mush, basically. The way that we deal with the jellies is we try and filter them out. So actually, when we, because we're focused on the mesozooplankton, we're really trying to get rid of the larger zooplankton, which tends to be the jellies. And so when we get a sample, we basically rinse it into a bucket. If we see a lot of jellies in it, we rinse it through a really coarse mesh, basically like a dive bag. And we take all the large things, it could be jellies, could be fish. I think we've gotten some random crabs. We throw those back in. So we're not trying to sample those right now. But it's absolutely an important part of the ecosystem that I think we should be paying attention to.
A lot of the gelatinous zooplankton don't preserve well
Right.
either, so you can't even, you collect them and then they turn to
Yeah.
mush.
I mean, the other thing that people do, and that I was thinking about, is we could try and use like an eDNA method to sample them. So eDNA is basically DNA that organisms release into the water. People define it in different ways, but you could think of it as usually things that are outside of their cells, just kind of sloughed off. The problem, it's a exciting but challenging method. One of the challenges is that it's not necessarily quantitative.
Yeah.
So, you could say, oh, there's a million sea nettles. There's sea nettles in the water, but how many? Like, I don't know. The way that it could be potentially useful is to get an idea of if there's a strong seasonality to it. Oh, we first detected it in April this year. Oh, we first detected it in February this year. So eDNA could be useful in that context. Yeah.
How can I begin to get involved with, buoy and marine biological laboratory?
Yeah. Yeah, so we do have a volunteer program. So that would be through our exhibit center, I think. If you want to, so I mean, if you want to talk to me afterwards, I can give you my email and put you in contact with them. We don't really have an active citizen science program, but we do have lots of ways that people can volunteer more in the outreach side of it. But I'd be happy to talk to you afterwards for sure. Yeah. Oh, I have that.
You mentioned using machine learning, to like identify zooplankton.
Yeah.
Are you constructing the data set yourself?
Yeah.
Or are you sourcing that from somewhere else?
Yeah, so there are ongoing community efforts for this. So the one that's really emerging for zooplankton is something called ecotaxa. And it's a big community effort to do this. The reason we're having to do a lot of it ourselves is because there are different types of instruments that people use to image the zooplankton. And the flow cam that we're using has been less used for zooplankton imaging. So there's not a big data set of annotated zooscan. Sorry, there isn't a big annotated data set of flow cam images. And so we probably will have to build that up ourselves. What we're thinking to do, one of the challenges with that is as the images are going through the instrument, they're at all different orientations, right? And some of them aren't focused and some of them aren't, and some of them, they can be jumbled up. So some of them might be easy to identify and some of them will be harder. And so one idea is you can just get better at it. But another idea that what we're working on is taking a sample and manually sorting it so that we have a little set of Calanus, a little set of Syntropodes, a little set of Acartia, whatever. And then when we have things where we know what they are, we can run that curated set through the flow cam over and over. So we get images of things that we know what they are, that are photographed from all different orientations. So that's what we're planning to do. And we're going to start doing that with we've done some of this work in Waquoit, but we also have some samples farther offshore in the Gulf of Maine. And we're going to start with those Gulf of Maine samples and sort them and run them through the flow cam and build up our annotation that way. Yeah, it should be really cool once we make some progress on it. Yeah. And we'll be curious to see, once we have that sort of improved annotation, sort of what proportion of the images the computer can really classify to a fine taxonomic level. One of the things that's frustrating to me is the way the tool is currently set up. It works best if you're able to give everything a name immediately. And what I would prefer to do is sort them into rough bins first. This is a copepod, this is a copepod, these are pteropods or whatever. and then within that classify. But that's not in terms of the way the software is set up, that's not the best thing to do. So it would be better if we could really classify things as finely as possible from the beginning. So that's what we're trying to do.
We've got a flow cam question in the chat.
Oh, boy, okay.
And can the flow cam detect and quantify gelatinous zooplankton?
It theoretically could if they were small enough. So our flow cam, organisms have to be smaller than 5 millimeters. So very, very small gelatinous zooplankton, it theoretically could, but otherwise, no.
So who would be the biggest users of the results that you're going to get?
Yeah, I think ultimately, perhaps the fishing community, as we sort of think about how this could affect fish populations, perhaps people interested in natural shellfish populations as we look at, so we can see shellfish larvae. I think also in terms of Waquoit Bay, just anyone interested in the effects of our human influence on the water quality. So as we look at sewage and septic input, either in Waquoit or other estuaries around the Cape, how changes in the water quality are affecting the marine community. So I think those are some of the main reasons that we care about it. Some of the main impacts. Yeah.
So you mentioned a early 2000s paper.
Yeah.
And is there anything even earlier than that? And also, are there old jars of alcohol that, old samples that
Yeah.
you might be able to process now using new methods?
That's a great idea. I haven't asked around about that. That's a really good idea and I'm not sure. Yeah.
Well, that makes me think of Arthur Hughes, who was with the old Boston University Marine Program at MBL.
Yeah.
In the 1970s, 1980s, Dr. Hughes specialized in copepod studies.
Okay.
So his papers might be of value. He had a grad student at the time named Tom Duncan,
Okay.
who has since retired from a career teaching biology. I know that Tom was sampling copepods in Childs River and in other locations nearby, specifically copepods in the 1970s.
Yeah.
And Tom's still around locally, so he might be a source of information.
Yeah. We'll look that up for
Is he
sure.
a pack rat? So is his garage boxes of jars?
People are reluctant to throw away samples.
I know, which is like, now we realize what a good thing that is.
Yeah.
Yeah,
I hope
absolutely.
it fights.
Yeah, it's really cool. Yeah.
Calamus, I can imagine how in times of poor conditioning and nutrition, they could shed fat and it would be easier to drop down in the water column. But overall, I'm wondering how individuals and populations of calamus can rise, fall, and suspend where they need to be, and how they do it and what drives that.
Yeah, so that is sort of debated. People have opinions about it. So there's interesting things about it. So one of the things is the oil sac so a lot of the fat, most animals store a lot of their fat as triglyceride. Calanus stores a lot of it as wax ester, and which is just a different kind of fat molecule. But one of the things that's interesting about it is as wax ester gets under pressure, there's a point at which it becomes more solid. And so then there's a phase change and that affects the density. There are also other interesting things that copepods that undergo that dormancy in the middle of the water tend to accumulate. I can't remember which direction it is, but they tend to change the type of cations in their blood, in their hemolymph, which affects their density. That's another thing. Trying to think, there's another adaptation, but there are several very specific adaptations that seem to affect their buoyancy. And something that, see, now you've given me permission to talk about my favorite topic. One of the fascinating things about that dormancy is nobody can recreate it reliably in the lab, which means we don't know exactly what controls it, which is fascinating, right? So people have tried manipulating light, temperature, food, sort of predators, sort of.
Pressure.
They have not. But we are building pressure chambers. So that is my secret plan. So, I mean, that gets right at what you're talking about that buoyancy. So I think that's a real secret to working with colonists. So stay tuned. I think it's going to be really cool. So our plan is to do more work with predator cues, play around with the food availability, but I think pressure is probably the secret sauce. So that's what we're going to try. Yeah. Yeah.
Question in the chat is the copepod species composition stable or changing?
We don't know. We don't know. So that's exactly what we'd like to know. And, you know, comparing it, the samples that we have aren't exactly the same as what they did back in 2004, because that wasn't really the goal of our study. But we are hoping to do ongoing sampling with our partners at Webinar so we can understand that. So that's a great question. So we'd like to know.
I have a comment from David who says, first, thank you, Anne, for explaining the topic so well. And secondly, I am going to be jumping this year from 3 years of Child River water quality monitoring with Ryan Clark, our stewardship coordinator here, to the Bay Watchers this coming season with Theo and you. And this was a helpful explanation.
Fantastic. I mean, we're so excited that the Baywatchers data is available and that people do that work, and it's a fantastic program. So yeah, thank you, Dave. I think
I have
Okay.
a question.
Okay, yeah.
Also from Jeff, from before, eelgrass has decreased dramatically since 1990.
Yeah.
Is there an effect on zooplankton?
We don't know if there's an effect on zooplankton. We certainly think there's an effect on the benthic animals. And some of those benthic animals, of course, have their meroplankton, so they have larvae. But we don't know how the eelgrass affects the zooplankton community. The eelgrass has certainly been changing because there are overall changes in the nature of the bay. So I would expect that the zooplankton community has changed. But unfortunately, we just started monitoring last year, so we just don't really have the time series to be able to answer that. It's a good question, though. It's an important question.
He asks, I don't know if I'll pronounce this right, Eurytemora species present?
Eurytemora.
Yes. Is that species present?
There definitely are Eurytemora, yeah.
Well, I mean,
You found some.
yeah, well, I've found them with whole. I don't know if we've ever seen them here.
Yeah, yeah.
But they're very likely.
Yeah, I mean, in the published, the work by Valiella et al., they studied, I mean,
There
there's
must be.
Eurytemora and,
Yeah.
yeah, they're definitely.
They're very good.
Yeah. Are Eurytemora a special favorite of yours, Jeff?
He did also comment / question. Arthur Hume studied parasitic copepods, a fascinating topic, and he's the world's expert. Question: did he study the local zooplankton?
We were just talking about, so apparently, yes, yeah. And we did see some of the parasitic species too in some of our samples. We weren't specifically studying them, but we see them swimming around.
That's all the online questions.
Yeah.
You mentioned using eDNA to detect presence in
Yeah.
different times.
Yeah.
Is there any thought of using it to look at the broader biodiversity of the?
Someone certainly could do that. We don't.
Yeah.
We're not doing that right now, but you could definitely do that. Yeah, to look at what species are present. Yeah, absolutely. It'd be a great thing to do. And even so, some of these copepods even have cryptic species, and so it would be helpful in trying to sort it out. So that figure I was showing you from the Chesapeake Bay, where there were like 2 species of copepods, that thing is a species complex, so lots of species, yeah. So you could sort some of that out, of course, with molecular methods. Yeah, okay.
I wanted to learn more about zooplankton. Giving suggested ways I do that.
Talk to Phil. I mean, so you're definitely welcome to come by my lab. And one place that we do some outreach is through, for example, the Woods Hole Science Stroll. So we have a table where we show some of our zooplankton. We can bring live zooplankton. That's one way. How else would you learn about zooplankton? Got any ideas, Phil?
The exhibit center, they've got stuff there.
Yeah, the Woods Hole Hui Exhibit Center would be a
Well,
place.
the way we learned about it is reading papers,
Yeah, absolutely, absolutely.
journals.
Yeah, zooplankton, they're a little underappreciated sometimes. So there aren't any like hard-hitting documentaries yet about zooplankton.
Books in that, it's not in there.
Yeah, there is a really nice documentary about the Gulf of Maine, that's recent. I don't know who put that on. Well, I'll think about that and I can get back to you. Anyway, thank you for very good questions, some information for me to think about, and it was really nice talking to all you guys tonight. Thank you.
Thank you, everyone, for joining.
That's Beth.